Klebsiella are gram-negative rod, non-spore forming rods, facultative anaerobes, oxidase negative. The question is, what is oxidase? An oxidase is an enzyme that catalyzes an oxidation reduction reaction, especially one involving dioxygen as the electron acceptor. In a reaction involving donation of a hydrogen atom, oxygen is reduced to water or hydrogen peroxide. Klebsiella, also lactose fermenter, form pink-colored colonies on McConkey agar. Presence of polysaccharide capsule in the culture plate. Mucoid colonies are seen. Non-motile. They can be found singly, in pairs, or in chains. This slide shows the disease that's caused by Klebsiella. Klebsiella are found in the human nose, mouth, and gastrointestinal tract as normal flora. However, it behaves as an opportunistic human pathogen, causing pneumonia, urinary tract infection, sepsis, meningitis, diarrhea, and soft tissue infection. Klebsiella species have also been implicated in the pathogenesis of ankylosing spondylitis and other spondyloarthropathies. The route of transmission. Acquired Klebsiella are acquired in the hospital setting or in long-term care facilities. In fact, Klebsiella account for up to 8% of all hospital-acquired infection. People with a compromised immune system and or people who have an implanted medical device, such as a urinary catheter or airway tube, are more at risk for Klebsiella infection. Extensive use of antibiotics has resulted in the development of antibiotic-resistant strain of Klebsiella. What is the treatment available for Klebsiella infection? Because the antibiotic resistance of these organisms, the choice of a drug depends on the result of sensitivity testing. Isolated from hospital-acquired infection are frequently resistant to multiple antibiotics. An aminoglycoside, example gentamicin and cephalosporin, example cefotaxime, are used empirically until the results of testing are known. Okay, let's move to talk about the second example of enteric organisms that cause an infection outside enteric tract, which is proteus. And let's start with characteristic features of proteus. Proteus are gram-negative, rods, motile, aerobic, and facultatively anaerobic. Urease positive, and we explain what urease do to the body when excreted by bacteria or other microorganisms. A striking microbiologic characteristic of Proteus species is their swarming activity. And let me explain what is swarming activity. Swarming phenomenon appears macroscopically as a concentric ring of growth emanating from a single colony or inoculum. On a cellular level, swarming results from bacterial transformation from swimmer cells in broth to swarmer cells on a surface, such as a gutter, in a process involving cellular elongation and increased flagellin synthesis. There are many species of Proteus. These are Proteus mirabilis, Proteus vulgaris, Proteus bineri, Proteus maxophysian, and Proteus hauseri, and 3 unnamed genomospecies, Proteus genomospecies 4, 5, and 6. What is the disease caused by Proteus? This slide shows the diseases that are caused by one species of Proteus, which is Proteus mirabilis. Proteus mirabilis is a common cause of both community-acquired and catheter-associated UTI, urinary tract infection, cystitis, pyelonephritis, prostatitis, wound infection, and burn infection, and occasionally causes respiratory tract infection, chronic suppurative otitis media, eye infection, meningitis and meningoencephalitis, bacteremia following catheter-associated urinary tract infection, cellulitis, endocarditis, mastoiditis, empyema, and osteomyelitis. Proteus mirabilis could have a role in the etiology of rheumatoid arthritis. Now this slide shows the disease that caused by other Proteus species. There are so many diseases caused by other group of Proteus. These are urinary tract infection, wound infection, burn infection, bloodstream infection, and respiratory tract infection. Proteus vulgaris causing bacteremia and brain abscess. Proteus myxophysian was originally isolated from a gypsy moth and has been isolated from urinary tract infection in India. Proteus hauseri has not been associated with infections in humans. After we know all the keys of these enemies, let us know what is the treatment that's available now and used against this species. Most strains of Proteus are sensitive to aminoglycoside and trimethoprim, sulfamethoxazole. But because individual isolates can vary, antibiotic sensitivity tests should be performed. Proteus mirabilis is the species most frequently sensitive to ampicillin. The treatment of choice for indole-positive species is a cephalosporin, example cefotaxime. Now I am going to explain some routine techniques that used in the lab to diagnose Enterobacteriaceae. First, culture on McConkey agar. McConkey agar is selective and differential media. Selective for gram-negative bacteria and used to differentiate Enterobacteriaceae that able to ferment lactose from Enterobacteriaceae that unable to or non-lactose fermenter. Lactose fermenter isolate will change or produce colored colonies, red or pink, while non-lactose fermenter isolate produce colorless colonies. So, Salmonella and Shigella and Proteus produce colorless smooth colonies because they are non-lactose fermenter. While Klebsiella forms mucoid pink colored colonies due to the presence of polysaccharide capsule and the pink color because Klebsiella lactose fermenter. Another technique that used in the diagnosis of Enterobacteriaceae, and I mentioned it in this lecture, is sugar fermentation. For testing sugar fermentation, like lactose, sucrose, and glucose, we use culture media called TSI, triple sugar ion, which is a semi-solid media having slant and bottom. This media contains 0.1% glucose. So if only glucose is fermented, only enough acid is produced to turn the bottom yellow. The slant will remain red. The second ingredient that involved in this culture media is 1% of lactose and 1% of sucrose. If lactose or sucrose or both sugar are fermented, a large amount of acid will produce, which turns both slant and bottom yellow. So the appearance of yellow color in both slant and bottom indicate that the isolate has the ability to ferment lactose or sucrose or both. Third, substrate is iron or ferrous sulfate, which is used as indicator of H2S formation. Fourth, phenol red, used as an indicator of acidification. It is yellow in acidic condition and red under alkaline condition. This culture also contains peptone, which act as a source of nitrogen. Note, remember that whenever peptone is utilized under aerobic condition, ammonia is produced. Let's move to explain the procedure. This figure shows how the triple sugar iron looks like in the tube. And the top is the slant and the bottom. Procedure include several steps. First one, with a sterile straight inoculation needle. So that touch the top of a well isolated colony. So to culture bacteria on triple sugar iron, we use the needle, sterile needles. Then inoculate triple sugar iron agar by first stepping through the center of the medium to the bottom of the tube, and then streaking on the surface of the agar slant. Stepping means الطعن. Last step is leave the cap on loosely and incubate the tube at 35 to 37 in ambient air for 18 to 24 hours. This slide shows you the triple sugar agar cultured with different species of bacteria to show you how the result will be expected. So the origin media have pink color. If the growth or bacterial growth result an acid slant and acid bottom, this means the bacteria ferment the sugar and the acid will change the color of media, make it yellow slant, yellow bottom. So yellow slant, yellow bottom indicate to the fermentation of glucose, lactose, or sucrose. If the result alkaline, mean red slant, red bottom reaction. So this means absence of carbohydrate fermentation. Black color in the third tube indicate the formation of H2S. The space that appear in the fourth tube indicate to the gas production. Let's move to talk about motility tests. This slide gives you some information about motility tests such as culture media and method. So the culture media that used to test the motility test of bacteria is a semi-solid media named SIM medium, sulfide indole motility medium, which is a combination differential medium that tests three different parameters: sulfur reduction, indole production, and motility. In SIM, motile bacteria swarm and give a diffuse spreading growth that is easily recognized by the naked eye. The procedure for testing the motility involved attach a straight needle to a colony of a young 18 to 24 hours culture growing on agar medium. Stab once to a depth of only 1/3 to 1/2 inch in the middle of the tube. Be sure to keep the needle in the same line it entered as it removes from the medium. Third, incubate at 35 to 37 centigrade and examine daily for up to 7 days. Observe for a diffuse zone of growth, flaring out from the line of inoculation. So what is the expected result after 7 days? The expected results of motility test, if it's positive, so there is a diffuse hazy growth that spread throughout the medium, rendering it slightly opaque. While the negative results mean the growth that is confined to the stab line, with hardly defined margin and leaving the surrounding medium clearly transparent. This slide shows you another routine use or test that's used to diagnosis of Enterobacteriaceae, which is urease test. The culture media that is used is urea agar base, or name Christensen urea agar. What is the benefit of urease? Urease analyze the urea, as I said previously, to ammonia and CO2. The presence of ammonia change or made the medium alkaline. The uses of urease test. First, to test the ability of an organism to produce the enzyme urease that hydrolyzes urea. And second, to differentiate urease positive protease from other Enterobacteriaceae. In routine diagnostic laboratories, the urease test result is read within 24 hours. If organism produces urease enzyme, the color of the slant change from light orange to magenta. If organisms do not produce urease, the agar slant and bottom remain light orange, medium retain original color. This slide and the one after supply you with a guide of expected results for triple sugar iron agar or sugar fermentation test, motility and urease test for some of Enterobacteriaceae bacteria, such as Salmonella typhi, Salmonella paratyphi A, Salmonella and other Salmonella species, E. coli, Klebsiella, Shigella, and Proteus. This one also the same, and show you how to read this slide. So, Salmonella enterica, the growth on triple sugar iron will give you red slant, mean alkaline slant, yellow bottom, mean acid bottom, with gas, positive formation of gas, and black bottom, which means produce of H2S, etc. After listening to this lecture, you would be able to answer these questions. So, read them carefully and try to answer them. Good luck. Thank you for your listening. Please don't hesitate to ask me any question about this lecture.